Excellent. It was a lot of fun.
Let me introduce you to our speaker. This is Professor John Papanikolas, professor from...
Nice to meet you.
Great. Glad you're here. Oh, he's going to do it.
First, I want to thank Dan for inviting me, and thank you all for turning out. What I'm going to tell you about today is not actually part of our EFRC work. It's another project that's going on in my lab. What we've been trying to do is combine ultrafast spectroscopy with microscopy to develop techniques where you could image carrier dynamics with both spatial and temporal resolution. And before I get started, I really need to thank the people that did the work. It started out with a few students in my group. Brian Mell and Ralph House were really the ones that really started the project, and they've since moved on. Justin came along sort of in the second wave, and he and Michelle did a lot of the work that I'm going to tell you about today on the silicon nanowires. And Justin is now at Intel, and Michelle is kind of sort of a senior student. And I got a kind of the third generation of students coming along. Emma Kading has sort of started in the last couple of years, and she's been contributing as well. And then there have been a couple of postdocs that have contributed. But beyond, in addition to my group and the talented people that have participated in the project, I really got to say I'd be nowhere without sort of the help of people that actually make the materials. And the physical chemistry and the things that we can do and the sophistication of the questions that we can ask and answer really depends on the quality of the materials. And so I've been really fortunate to sort of have this collaboration with Jim Cahoon, who's an assistant professor in our department. Started a few years ago and is an expert in sort of growing nanowires. And he and his students have worked closely with us in sort of making the materials and providing us with some really, quite exciting systems to study. And this is kind of sort of where I'm going to start. This is an SEM micrograph of a bunch of silicon nanowires. These all came from Jim's group. And this circle that's up here is about the size of a laser spot that you would have in sort of a conventional pump probe experiment. And when you do an experiment with a laser spot that size, not surprisingly, you're averaging over a huge number of structures. All of these little wispy-looking things in here are the silicon nanowires. And that's fine if you're at this altitude and you're kind of looking at it and it's, everything's great because it all looks good. But as you start zooming in, what you realize is that there's a lot of heterogeneity in the sample. You got different sizes, you got different shapes of wires. Some are straight, some are bent. There's also a lot of other stuff in here, amorphous silicon, who knows what, that would also be contributing your signals. And if you zoom even further, you start to see sort of that there's the catalyst particles, there's stuff that's not even real nanowires, and so on. And all of this stuff contributes to your signals in sort of conventional spectroscopies. And so the question that we had was, could we do an experiment where we sort of excite a particular part of a structure, like the end of the wire or the middle of a wire, and measure the dynamics or the carrier dynamics at different points within a single structure? Or could we develop an experiment where we could excite in one place and probe at another and watch carriers move, created in one location, end up in another location? And so that meant sort of developing new types of pump probe microscopies. And so you can kind of organize those on kind of a diagram where I got sort of the spatial resolution and the time resolution here. And if you're thinking about spatial resolution, I don't really have to sort of talk about STM and AFM here, but they're like sort of the gold standard in your ability to sort of resolve things spatially. And if you want to do near field optical spectroscopy, you can do that. It's still pretty high spatial resolution, but all these techniques have kind of low time resolution. You go to confocal, that's easier to sort of manipulate again, still sort of slow time response. If you want sort of fast time response, you can do time resolved emission. There's a gazillion techniques to do this. You got fast time response, but you have no spatial resolution. And if you really want fast time response, you could do pump probe, and that's kind of the gold standard of sort of ultrafast spectroscopy. It gives you the highest time resolution where it's determined by the laser pulse. But again, you have no spatial resolution. So the place you really want to be is in this part of the diagram. And early on, people sort of recognized this in the 90s and kind of the early 2000s, started combining sort of time resolved emission methods with microscopy. And there are a lot of examples of this, especially in biology and other fields as well. And you can get sort of roughly 50 picosecond or so time resolution without spending, a huge amount of money. You could do faster if you spent more money, but you're really limited by your detection electronics and the way you detect the light. But you're also limited in that you can only look at things that emit light. So if you wanted to look at something like silicon, which doesn't emit any light, you can use these sorts of techniques. And so where you really want to be, or where we wanted to be, was over in this part of the diagram, where we're looking at sort of pump probe techniques combined with some sort of microscopy. And if you went out and looked in the literature, and what you'd find is that, okay, we're not the first people to do this. There have been papers that have appeared over the last 15 or so years. In the beginning, they were all sort of low spatial resolution. They take their laser pulse, focus it with a microscope objective down to maybe, 5 microns or 50 microns, look at single particles and that sort of thing. There was no imaging involved in it, although they were doing sort of single particle type measurements. Later, my group and others started pushing that spatial resolution down to the diffraction limit, and that's where we're working. And there were several others that are doing this as well. This part of the diagram way down here, okay, that would be really cool to do, sort of like subdiffraction limit. So, it requires, I think, in a lot of respects, sort of working this area as a science in and of itself. Understanding sort of how the light interacts with nanostructures, and then sort of exploiting that to do microscopy. And it's a very exciting field, but like I said, it's almost just the process of localizing those fields and so on is almost a science of in itself. And so we sort of backed up and said, okay, let's try and do sort of conventional microscopy, but do pump probe spectroscopy in the microscope. And so we've been sort of working in this range. So our spatial resolution is a few hundred nanometers. Time resolution is a few hundred femtoseconds. And so we've tried to pick problems that kind of match those limitations. And so the goal was to take a sample like this, these are silicon nanowires, and do this pump probe microscopy and ask questions like, do the dynamics depend upon where you excite in the structure? If I excite near the middle of a structure or towards the end of it, does it depend on whether the object is straight or is it bent, and so on? And can we look at motion of charge carriers through materials? And so I'm going to start here with just telling you about the instrument itself. The first part is basically a conventional pump probe spectrometer in a sense, right? We got a Ti-Sapphire oscillator. We have an OPO, so we can do 2 color experiments. We get 2 beams. One's called the pump beam. That's the thing that excites the sample. We have our probe beam. That comes along later at some well-defined time delay. Those 2 beams get recombined, and they travel into an objective, which focuses them down to a diffraction-limited spot. And they're overlapped with some structure in our sample. And we then take the probe beam, collect it after the sample, and do lock-in detection. And essentially the type of technique I'm going to tell you about today is transient absorption or transient transmission experiments, where we're looking at the change in intensity of the probe beam as a function of whether or not the pump is there, okay? Or how the intensity of the probe beam changes with the presence of the pump. This is sort of an example of our spatial resolution. With 800 nanometer light, we can get somewhere down around 400 nanometer sort of spatial resolution in a 2-photon excitation mode. If I did this with single photon with 400, that's about where we'd be as well. This is the time resolution of the instrument. This was measured at the sample, so we get about 500 femtosecond sort of temporal resolution. We could do better than this if we put in prism compressors, but for a lot of the stuff I'm going to tell you about today, that the time resolution this is perfectly adequate. In our microscope, we could also combine our measurements with time-resolved emission imaging just by collecting the emission from the sample and detecting it. Although I'm going to only tell you about silicon today, so this part isn't gonna come into play too much. So this is basically the idea of the experiment, and that I'm going to tell you about for all the stuff that I'm going to talk about today, just in sort of different forms and configurations. It's a 2-pulse laser pulse technique. We have a pump pulse that's tuned to 425 nanometers, which is above the band gap of silicon. The beam is going to be focused down to a spot size that's about the size of that circle. So here's a nanowire. It's about the nanowire. That nanowire is probably about 100 nanometers in diameter or so, maybe 150. That thing there is the gold catalyst that was used to grow the nanowire. And so our pump pulse is going to be small compared to the length of the nanowire, but it's going to be large compared to the diameter. And in that region where it excites the sample, it's going to create a whole bunch of electron-hole pairs, somewhere in the neighborhood of maybe 50 or 100,000 electron-hole pairs in that focused spot. Pump pulse is going to go away, and then we're going to come along later with a probe pulse that's at 850 nanometers, so it's out in the near infrared. And in silicon, it turns out that when you put carriers in the conduction band or you photoexcite silicon, it becomes more transparent. And so what will end up happening to this probe beam is a little bit more of the probe beam will pass through the sample and be detected by our detector. So what we're going to do is look at the change in intensity of the probe beam with and without the pump. And what's going to happen is when you have carriers in the conduction band, the intensity of the probe beam is going to go up a small amount. Okay, so here's an example. So we took a nanowire. This is a silicon nanowire, and one of the things that we can do is we can take the samples out of our optical microscope. We can go over to the electron microscope, find the exact same wire that we used to do the optical experiment. So this SEM is the wire that was used to collect this data, and the place where the red circle is the point in the structure that we excited. Okay, and so what I'm plotting here is the change in intensity of the probe light as a function of that pump probe delay. And so initially at t equals 0, you see a big increase in the intensity of the probe beam. And then over time, as those electrons and holes recombine with each other, that transparency goes away. And it actually turns, in long times, it turns into a slight absorptive component, which we think is either has to do with trapped carriers is one possibility. Another possibility would be that it has to do with thermal changes or thermal effects. But whatever it is, this long-lived absorptive signal, it's there, and it lives for a long time, and in some respects I'm not really interested in it. What I'm really interested in is sort of the decay of this sort of fast component. And that decay reflects the recombination time of those electrons with the holes. And this can happen through lots of mechanisms. It could be sort of Shockley-Weed-Hall, Auger recombination. It also can take place at the surfaces. And in nanostructures, that tends to be sort of the dominant mechanism, but we could sort of test that because what it should do is as those carriers recombine at the surface, that lifetime should depend on the diameter of the nanowire. So if it is a surface-mediated event, it should depend on the lifetime should depend on the diameter, and it's inversely proportional to this thing called the surface recombination velocity, which is a metric of the quality of the surface. But anyway, so we could pick a different wire. So here's another wire. It's larger diameter, and sure enough, it sort of slows down, suggesting or consistent with the electron-hole recombination being dominated by the surfaces. There's another source or contribution to the decay of this signal, and that has to do with the fact that when I create all these carriers, they can actually move out of the volume of the excitation volume. And so even though they haven't recombined, they can still sort of lead to a decay in that signal. And so that gets kind of the second type of experiment that we've done, which is to actually look at sort of this carrier motion. And so here's the idea. We're going to excite with our pump pulse. We're going to wait. Carriers are going to move away, and then we're going to come in with our probe pulse sort of offset with respect to the pump and detect the arrival of carriers as they come into this location. The way we do it is it turns out in this microscope, if I bring my pump and my probe in exactly collinear to each other, they're going to focus in exactly the same spatial spot. But if I bring the probe in at an angle relative to the pump, its focus is going to be offset laterally. And so I can control sort of the position or scan the probe relative to the pump simply by scanning the angle that the probe beam enters the objective. And so we do that with a couple of mirrors that basically sort of scan that angle while keeping the beam sort of centered on the back of the objective. And we can image with it. This is UNC's old well, and it's their icon, I guess. And you can tell it's old because it's cracked. But we have this sort of little logo, and we can sort of scan it with, this was an image we took with the stage, which the sample scanning XY stage. And this is what we get with our mirror assembly. So we can scan sort of distortion-free across 10 or 30 microns or so. And we can do it with very little, there's almost no difference in the pump probe delay across that image. So if you've done ultrafast spectroscopy, you know that as soon as you introduce path length into your beam, you're going to introduce delay. But it turns out that in this particular case, geometry is on your side, because whatever path length you introduce by tuning one mirror is taken up by the other. And so there's almost essentially, as near as we can tell, no time delay difference across the image. Okay, so what we're going to do is the following experiment. We're going to take a nanowire, a silicon nanowire. We're going to pump it at that point, and our pump beam is going to stay fixed in this experiment. Then we're going to fix the probe beam, the delay between the pump and the probe at some time, 0 picoseconds, 20 picoseconds, something like that. And with that fixed delay, we're going to take our probe and we're going to raster scan it over the pump. And the idea is that, so here's the image that you get at 0 picoseconds when the pump and the probe are both overlapped in the wire. Green in this color scale corresponds to like 0 signal. So when my pump and my probe are way separated from each other, I have no absorption, which is a good thing. This like light thing here that's a representation of where the nanowire is. And then this red signal, the red in these images corresponds to increased transparency. So this image that you have here is essentially a spatial map of the size of the transparency created by the pump spot. Okay, and it's larger in diameter than the nanowire because our spatial resolution is about 300 to 400 nanometers, and the nanowire is only about 100 nanometers. So then if I essentially take my time delay and now collect images like this at longer delays, what you see is that over time, as the delay gets longer and longer, and as carriers move away from the excitation spot, that transparency also moves with it. And so what you're looking at is the evolution of that photo-induced transparency spatially, which is also telling you about where the carriers go. And it's going to cycle through here and again. So it grows along the nanowire. It's decaying in intensity. So that's what this little this scaling factor tells you. So the whole image is getting short, coming down in intensity and sort of spreading out. The blue thing that ends up in the end is in the latter frames is that of photo-induced absorption, and that's there always at long times. But what's interesting is you can actually see that it sort of spreads as well, which led us to think that it had to do with trap carriers, but it could also be sort of thermal effects that were exciting the wire and heating up a small section of it, and then that sort of spreads along the wire as well. So we can sort of directly image this carrier motion. It turns out that these images are great and they're a lot of fun to make, but you can't really do anything quantitative with them. And so there's another mode you can do. Oh, and okay, so if you want to do the old school side. So there's a whole set of all those images just on one slide. We've done it in multiple wires. In some wires, in this particular one, we don't see the diffusion, which has to do with the fact that I think the surfaces are really lousy, and so the carriers don't have time to go anywhere. So if you actually look at, this whole time frame here is about 50 picoseconds compared to the hundreds of picoseconds up here. But nevertheless, we see this in multiple wires. But it's not. But these types of images aren't. You can't do anything quantitative with like sort of pulling out sort of carrier diffusion times and so on. And so we can do a different type of experiment where now instead of what we're going to do is hold the probe beam, the position of the probe beam fixed. So it could either be spatially overlapped with the pump, or that's where that blue circle is, or it could be at one of these remote locations. And then we're going to scan the delay time. And so when they're spatially overlapped, you see this sort of really fast decay. It's essentially the same thing I showed you on the previous slide. But when they're when the overlap is larger, you see this delayed rise. And so here's like, with B and C and D, that's like we've got 4 extras here. There's E, you can barely see it. Turns out there's another one that's way down in the noise. So we're actually able to pull out with pretty high signal noise sort of the arrival of carriers at remote locations. And then you can say, okay, well, it turns out, how does this compare with sort of bulk silicon? So you do sort of a simple diffusion model with bulk silicon parameters, and this is what you get. And there are some small differences, but for generally speaking, the carrier motion in these nanowires is more or less consistent with carrier diffusion in sort of bulk silicon, which isn't all that surprising. we've gotten a little more sophisticated since we took that that initial data, and now we actually have expressions where we can mathematically model sort of these decays, and so we can. So here's like intensity as of our signal as a function of time, and spatial separation. That's the delta x. And what this does is it basically combines sort of the diffusion equation with the single exponential decay, which has a lifetime tau. There are other, the diffusion constant enters into this equation, which isn't all that surprising. And then there are also parameters that have to do with the geometry of our experiment, which particularly the laser spot sizes and so on. But I can take this equation and simultaneously fit all these different transients that we get out of the experiment and extract a single lifetime. Okay, so even though every individual transient is highly non-exponential, there's really the population decay is described by a single time constant. And that time constant in this particular case is around 220 picoseconds. If I use that equation that I talked about earlier with that related to lifetime to surface recombination velocity, I can pull out a surface recombination velocity of about 5,700 centimeters per second. And all this stuff was fit with the bulk diffusion constant. Okay, so we can now, we don't have to like take transients at all these different spatial locations. I can get lifetime simply by just measuring sort of the decay at in the spatially overlap configuration. Then I can pull out, I can do this all day long, or my students can do it all day long. And so that's what I told them to do. That's what they volunteered to do. And so we took a sample. This is a sample of a bunch of nanowires. And what they did is they just went through and just looked at a whole bunch of different wires, like maybe ran the experiment maybe 200 times or something like that. And we pulled out a whole series of transients. That's what all these are. They're plotted on a log scale here, so but they're essentially they look exactly the same as the ones that I showed you earlier. And the first thing you get is that there's a wide distribution of sort of decay kinetics here, which isn't necessarily all that surprising, given sort of the heterogeneity in the sample. And we thought, well, what we could do is we could take that equation, we could fit them to that expression I had on the previous slide, pull out a series of lifetimes, and I can get a distribution of lifetimes, and that's what's given down here. And there's sort of an average surface recombination velocity of about 7,000 centimeters per second. And so, we felt pretty good about ourselves and everything, and then we thought, well, we can check this because we're not 100% sure that we're right. We could actually do the transient absorption experiment, which is the ensemble measurement. And the question is, could you obtain the same information in the conventional technique? And so in our lab, we also do transient absorption. So we took the same sample, exactly the same chip, and we put it into that spectrometer, excited at the same wavelength or nearly the same wavelengths. And this is what you get from that. And this is the ensemble measurement. And it looks, similar. These points here are actually those gray points, so they're a little bit faster than what you get from the microscopy. And but we thought, well, could we actually pull out a distribution? And because this decay is multi-exponential, so you could fit that decay sort of in this sort of way, where you have like a distribution of lifetimes and weighting all these single exponentials. And then I could take that and fit it to get this distribution. But if you've ever done anything like this, you know that those fits are like highly unstable and they never really work unless you know something about that distribution. And so, but we do know something about that distribution because I can measure the diameters. And so we took the chip and we went to the SEM and got the distribution of diameters. And that gave us, we could use that and then that kind of little formula that relates lifetime to diameter to get a distribution of lifetimes. And then I could use that in this expression, and when I do that and fit it, I get that distribution. So transient absorption gives me a distribution of lifetimes that's consistently, and then we did this on 4 different samples at different diameters and so on, and it's consistently always faster than what we get out of the microscopy. And so the question was, okay, what is that? And so at first I said, well, it has to do with, these samples that Jim gave us. There's all this amorphous stuff, and God knows what's in there, and maybe that's what's contributing. And Jim said, no, I don't think so. I guess he was a little sensitive. And so we started to think about what could be going on, and it turns out that yes, both experiments were done on exactly the same sample, but it turned out that in order to do the transient absorption, we had to do that in a part of the film that had a lot of nanowires, and because you had to have enough absorption intensity to measure it. And so that was in the center of the film, and this is what you would get there. That's what the SEM looked like from the center. But in order to do the microscopy, that was too many wires. They were overlapping. It was hard to make sense of what was going on. So we'd go over to the edges of the sample where there were fewer wires. And this is what the SEM of those wires looked like. And one thing you can tell, like just comparing these two things is, okay, here all the nanowires, or a lot of them, are straight, like just like you would expect them to be. And down here it kind of looks more like a bowl of spaghetti. There are a lot of curved wires. There are some straight ones, a lot of curved ones, and so on. And so we started to think, well, does that curvature have something to do with the differences that we're observing in the two techniques? Because in the transient absorption, you're averaging over all of this stuff, but in the microscopy, you're selectively sort of sampling just more or less the straight wires. And so we started looking at sort of these bent wires. So here's a bent wire, and if you measure the kinetics at all these different points, you get these blue curves. There's actually 3 transients there. They all overlay. But you look in the bent segments, and they're faster. And in fact, you can do sort of the transient absorption imaging in a slightly different mode. So this one is the pump and the probe are spatially overlapped with each other, but I'm going to drag them across the structure at a fixed time delay and get an image of what the transient absorption signal looks like across the structure. So at 0 picoseconds, it looks something like this. You would expect it to be sort of, or we might have hoped that it would be sort of uniform across the structure. It turns out that it's not, but we understand kind of why it's not. It has to do with the, mostly with polarization effects that have to do with sort of probability of exciting the wire, whether the excitation is parallel or perpendicular to the wire axis. So since we understand that, we can sort of, we can ignore it, or at least not sort of focus on that too much. And we can sit there and sort of scan images at different delays. So here's at 50 picoseconds, it gets dimmer. And at 180 picoseconds, you can sort of see that the bent region has gone away even more. But it's actually a lot easier to see if you normalize these images along the wire. And so we did that. So this is the same image, but all we did was we went along the wire, and whatever the intensity was, we divided sort of the intensity in that region by the intensity. So by the intensity at the max at 0 picoseconds. And so that this is all been along the wire. It all has the same intensity now. And now if you look at it, what you see is that sure enough, and it'll cycle around in here in a second, this bent region kind of goes away faster. And if you stare at it long enough, what you'll actually see is that bent region, it actually eats away at the image as it goes along. So it's not only are the bent regions faster, but there's a correlation between sort of the lifetime and the curvature of the wire. And this is in many wires. So here's another one. This was question mark wire. And then here's another one, does the same sort of thing. So everywhere we looked, wherever the wires were bent, the recombination kinetics were faster. And so we thought, okay, can we start to quantify this? And so we'd take the samples, and we know exactly what the structures are. And so at each point that we measured the kinetics, we would also sort of figure out what the radius of curvature was. And when you do that, you can see there's this sort of clear correlation between sort of the radius of curvature and the lifetime. And this slide's like 6 months old, and I got many more points that I could put on there, and the correlation is better. And it turns out, okay, so it turns out when you take this wire and you bend it, what you're actually doing is you're straining it. And so one side of the wire, it's in tension. On the other side, it's in compression. And you can quantify that strain with just a really simple formula. So if I know the radius of curvature and the diameter, I can get the strain. And so now I can put a strain axis up here. Okay, so at the highest or the largest or the most bent we had, at least in this data set, we were somewhere around 3 to 5% strain in the nanowire. This is essentially, it's more or less the same data, but now it's the rate of recombination, and it's on a linear scale here. This is with 100 nanometer wires. If you do it with 50 nanometer wires, it's a little bit different, which told us that whatever was going on was happening either at or near the surfaces. And so we started to think, okay, is this, are we just changing defects or what when we do this? It turns out that that's was our initial thought, that as we bent the wire, we were just introducing defects into it. But it turns out that there's some calculations by Grossman's group looking at sort of strain in nanowires and its effect on the band gaps of the valence band, the conduction band. And it turns out that that when you strain the nanowire, the band gap actually goes down by about a factor of 2. I mean, this is not a small effect. Over sort of similar ranges, of which this is like 4 or 5% strain, which is sort of similar ranges that we were seeing in our experiments. And what ends up happening is on one side of the wire, the valence band and the conduction band both go up in energy, but the conduction band goes up less, so the gap gets smaller. On the other side of the wire, they both go down in energy, but the conduction band goes down less, so that goes down more, so that the band gap gets smaller. And so maybe it could be that all we're doing is changing the electronic structure of the nanowire itself, so that if you imagine, okay, here's a nanowire going kind of in and out of the screen, and if it's straight, the band is going to be flat across the wire, and there are going to be a series of surface states and so on, and that's going to give me a certain amount of recombination. But then as I bent it, so you can imagine now the wire is kind of coming out and curving off that way. On one side of the wire, I'm going to be under compression. On the other side, I'm going to be under tension, and the bands are all going to shift. And so maybe we're introducing more surface states here, but maybe we're also just seeing sort of an effect of the change in electronic structure near the surface. And so we're trying to figure out. We've been writing this paper, and it's really kind of it's kind of getting to that part of the discussion where it's like, well, it could be this or it could be that, and so on. And so we tried to figure out if there was a way that we could actually figure out which one it was. And we thought, well, if the process was reversible, right? If I took the nanowire and squished it up, or measured its electron-hole recombination, and then squished it up, and it got faster, and then stretched it back out again, it got slower. So if it's reversible, then perhaps that's telling me that it's changes in band structure. Because I figured that as soon as I introduce defects into that system, it would be hard to get rid of them. Okay, so what we did is we took a piece of PDMS and we stretched it, and then we deposited all our nanowires on it and put that in our microscope and measured the kinetics along the wire. And then we let it go, and all the wires sort of squished up. And then we did the same experiment on the same point and the same wire, and then we stretched it back out, okay, to see if it was reversible. And so here is an example, and this data was taken. What is today? Thursday. So this was done on Tuesday. And so here's the wire before you did anything to it, and we measured the kinetics here, and that's this black curve. Then you sort of squish it up, and it gets kind of all bendy. And that's the second one, so it gets faster. And then sure enough, as you pull it apart, the recombination recovers and goes slow again. So it really looks like it's a reversible process. And it says probably what we're seeing is sort of changes, we're affecting sort of the band structure of the nanowire and not sort of introducing more defects into the nanowire. All right, so that was kind of fun. And all that stuff was on, these were all undoped wires, everything I was telling you about, just undoped silicon nanowires. One of the things that Jim's group can do is they can actually dope the silicon, and they can dope it N-type and P-type along the nanowire. And so we started looking at sort of carrier motion in these PIN junctions. So I'm not sure how much I have to talk about that here, but I made the slides. So if you take a P-type and N-type material and you bring them together, well, you end up in the junction between the P-type and the N-type material. You have this depletion region, and there's a field, a static field that extends across that junction. And so when you excite, this is how solar cells work. So everybody here knows all about this, and you get this charge separation event. So the idea was, could we actually image charge separation in these junctions? And so Jim's group has made things that are what's called PIN junctions, where you have an undoped layer in between the P-type and the N-type material. And all this really does is sort of extend the depletion region, makes it a little bit bigger. And his group has done sort of simulations on these things and modeled sort of charge densities and fields that you get in these junctions. But everything that they had sort of grown was like, the intrinsic layer was like 50 nanometers or something like that. And what we really wanted was something a little bit different. So they made us one where this intrinsic region was much larger. It's like 2 microns long. And so we did the simulations on these. it's a little bit different. You don't get complete sort of recombination of those mobile carriers. You actually get accumulation of holes at one side and electrons at the other side. So you have this sort of spikes in the electric field along the junction, but there is a non-zero sort of static field across much of the undoped layer. And so the idea was we wanted to do is like excite the middle of this junction and watch electrons go one way and holes go the other. These are the junctions themselves. We looked at a bunch of them I got data, or we looked at like 5 or 6 of them. Here are 3 different ones. At the end of the wire is the gold catalyst, and then we know based on how they grew them that there's like 3 microns of an N-type material, then 2 microns of an undoped material, and then there's a long section of P-type material. And we don't, when you look at the SEM micrographs, you don't know like where the doping is. But there is a slight change in the diameter at the interface between the P-type and the I-type, which has to do with the way the growth changes. And Jim's group understands this, and we were able to see it in the SEMs, and they were exactly where we thought they were going to be. And so we're fairly confident we know where the junctions are, or where the boundaries are between the different materials. Okay, so the first experiment we did was just to look at sort of the carrier lifetimes in these different regions. So if I do this sort of pump probe experiment in the P-type or the intrinsic undoped regions, you sort of see sort of similar decay kinetics. But if I look in the P-type region, it's much faster. And at first we thought that this really had to do with, like, maybe this was something sort of was kind of unique to this sample. This was the first time we really looked at sort of P-type silicon. But it turns out we've looked at lots of other P-type samples now, and they are just faster than, the recombination is just faster than the intrinsic or the N-type. And we're not 100% sure why that is. But nevertheless, it is faster. Okay, so this is an experiment where we take the pump and the probe, and they're spatially overlapped, and we're going to scan them across the sample. So it's essentially the same type of experiment, but now you can sort of see it in an imaging mode. So in the N-type region, you sort of see it decay, and in the intrinsic region, you see it decay slowly, but in the P-type region, it decays faster. What was interesting, and why I'm showing you this plot, is that the signal across the intrinsic region is not uniform. And in fact, near these boundaries, you have faster loss of signal. I'm not sure it's necessarily recombination, but you certainly see a faster loss of signal in those regions. And so we started to wonder if that was like due to this field that might be established at the boundary. But at this point, I think that's certainly consistent with, but that's kind of speculation. Okay, so anyway, what we did was we took this junction, we're going to excite it in the middle of the undoped region, and then we're going to look to see, do we see charge separation. And so this is one of those spatially separated images where the pump is always here in the middle of the intrinsic region, and then we're raster scanning the probe over the pump at a series of different time delays. And what you see is initially you sort of see this charge cloud expand. It reaches the edges of the intrinsic region, and then all of a sudden sort of a piece of it peels off and heads north. And then you're left with this long-lived trap signal that we can't seem to get away from, sort of appearing at longer times. With one other exception, or one other thing that I want to say is that in the last frame, which will like appear here in a second, there's a little bit of signal that's left even 12 nanoseconds after excitation, which is much longer than we've seen in any of the undoped wires. We think that that's this charge separated, or perhaps a charge separated state. So here are kind of 2 examples that we got, or 2 different wires. And again, you see sort of this signal that sort of expands, reaches the boundaries of the undoped segment, and then it sort of becomes asymmetric with a long-lived signal in the N-type region. And so on one hand, we were happy because the signals were asymmetric, but what we had envisioned was that, like, you create a bunch of electrons, and okay, this is our not knowing what we're talking about part. But you create a bunch of electrons, and what we thought would happen is they would just start marching across the intrinsic region in this field. But you don't see that. What you initially see is something that looks more like diffusion. And it's not until much later that you see sort of this, the effect of the junction sort of take over. And so we started to think about, okay, why is that? And so we've done these these finite element modeling of these systems. And we modeled our system as just sort of an intrinsic. We just took an undoped nanowire, and we applied a bias to it so that we don't have any electrodes in our experiment. But that's how we're going to get this sort of static field. And then in the center of the wire, we create a bunch of electron-hole pairs. And if I have a really low carrier density initially, like say 10 to the 15 per cubic centimeters, you see sort of the electrons move one direction, you see the holes move the other direction. It all looks exactly like you'd expect out of freshman physics, and everything's great. If you increase the density by about 2 orders of magnitude, now all of a sudden you don't see the electrons separate from the holes. So this purple shading is because the blue and the red sort of overlap. And so the electron distribution, the hole distribution in this image, or a series of images, are the same spatially along the wire. And you see a slight sort of shift of that packet towards the positive electrode, which in this case, in our experiment, would correspond with sort of the N-type region. It turns out that that shift is due to the differences in the mobilities between the electrons and the holes. Okay, and the reason why you don't see charge separation is because you have a whole bunch of electrons, you have a whole bunch of holes, and it takes energy to pull them apart. And that carrier density is too high, and the field in sort of the junction is too weak to overcome that. And so you actually don't see charge separation until the carrier density through recombination decays to a point that's below a threshold, and then you'll see it. And in fact, in these simulations, you'll see charge separation way out in the wings of the distribution where the density is really low. And so the thing I want to point out here is, okay, this carrier density in the simulations is 2 orders of magnitude lower than what we have in the experiment. Okay, so we're way beyond this point in our experiment. And so we thought, okay, can we actually model our actual images? And so we took that model that we had, essentially the same thing, but what we made one small change to it, and that was we put the surface recombination velocity in the section that would correspond to where the p-type material was. We made that 10 times faster than it was in the rest of on the rest of the wire, and because in computers you can do these sorts of things. And the idea was that this was going to account for the faster electron-hole recombination in that segment of the wire. And then we run the simulations, and okay, here are and from the simulations we get the carrier densities for electrons and holes along the nanowire. When we excite it in the center, these dashed lines represent sort of the width or the size of the undoped region that we have in our junctions, to kind of give you some scale. We would then take each of these profiles, and say the 600 picosecond one, and we would use that to give us sort of the density along the nanowire. And you can actually see it's hard to see in this image, but there is a little bit of charge separation that's going on in the wings here. But anyway, so we'd use that, and we could just convolve that with the Gaussian, and we could create an image that looks like sort of what the free carrier contribution to the images in our transient absorption experiment would be. We also have in the experiment this sort of absorptive component, which we can't get rid of. So we're going to just sort of add that in as kind of an ad hoc way. And we actually got the size and sort of the amplitude from the long time, or the frames that collected at really long delays in the transient absorption experiment. And if you add those two things together, you get something that starts to look very similar to what we have in our experiments. And so what we did is we did this for a whole bunch of different times, and this is what you get. So it starts out, and it sort of spreads through diffusion because the carrier density is so high that it could care less about the field. As that happens, some of the carriers sort of spill over into the N-type region. Some of them spill over into the P-type region, but they recombine so quickly in the P-type region that they essentially disappear. It's kind of like a little black hole. As soon as things go in there, they're gone. But in the N-type region, sort of the carriers spill into it, and then they decay over, hundreds of picoseconds out to about a nanosecond or so. What we don't see in the simulation is any intensity out here at really long times. And the reason for that is in the simulation we have these electrodes. So as soon as carriers are sort of separated or peeled out of that charge cloud, they're accelerated towards the electrodes, and then they're gone. So in the simulation, you can never build up a charge separated state. In the experiment, you can because there's no electrodes for or no place for the carriers to go. And so the vast majority of what we're seeing here is not necessarily charge separation. And in fact, we think from the simulations that probably only about 1% of the carriers actually undergo charge separation. But that a lot of the asymmetry has to do with the fact that you have these different sort of recombination velocities across the junction. Nevertheless, we do think that we are seeing at long times this charge separation. And so to kind of sort of go back to our data here, here's this kind of scheme I put up early on. At t equals 0, we come in and create a bunch of carriers sort of in the middle of this intrinsic region. Over the first 100 picoseconds or so, that charge cloud expands. By about 200 to 400 picoseconds, it starts interacting with sort of the mobile carriers that have accumulated near the boundaries. Spills over into the n-type and the p-type regions, but now on one side you have more holes, on the other side you have more electrons. In the p-type region you get very rapid recombination, so that signal all goes away. And then between 500 and 900 picoseconds you're left with you observe sort of the recombination in the n-type region, and then at really long times you have this charge separated state. We don't see sort of the signal from the holes, and that's kind of interesting. This could be maybe in the transient absorption experiment we're just not sensitive to the holes. That's one possibility. Another possibility is that maybe the holes get spread out over a much larger section of the nanowire, and so they're just their density is too low to observe. But anyway, I'm going to stop there and sort of thank all the people in my group that did this. I want to also thank the people that funded it. This was all funded by the National Science Foundation and the Army Research Office. And I do want to point out that I didn't use any EFRC money for this, since I know DOE is really sensitive to that. And so I want to certainly thank them, and I want to thank you all for sort of listening to me. And I'm happy to answer any questions. That's right. So it could. That's right, and so all I can say at this point is that the signals that we do observe appear to be sort of consistent with this surface recombination process, at least in terms of the geometry and how they depend on, or how they scale with diameter of the nanowires and so on. One possible reason for why we see such fast loss of signal in the p-type region could be because what we're actually observing is sort of trapping of the minority carriers there, which would be different than in the n-type.
And that can be also
That's right. We could start saturating traps and so on. All of the experiments that we did here, we're real careful to keep turning the laser down until we saw no change in the kinetics. And so we're down around about a picojoule per pulse in the microscope. So we really worked hard to make sure we weren't in sort of any regimes where we were seeing sort of nonlinear or kinetics that depended on intensity. And we are also, I mean, in part this was sort of a kind of a survival instinct, because as soon as the laser gets too high, you can start sort of melting the nanowires and so on. And we're comfortably below sort of that limit as well. So everything that I showed you, we worked real hard to be at places where the kinetics didn't depend on intensity, and also didn't change with time. So we weren't like introducing defects as we did the experiments.
We have some maybe a weak field in the PN junction, even in the intrinsic region, depending on the geometry. And in that case, from my experience in polymers, and I think in general, you have you carry a motion sort of spin the field. And this effect can give you the negative results, and this kind of thing. I can talk to you later about.
Yeah. I mean, I think that a lot of that is what we're seeing is that you could think about it as sort of there's an energy penalty pull electrons away from holes. You can also think about it as the charge distribution sort of moves to sort of screen the applied field, which in this case would be the field of the junction. I think those are all sort of the same, describing the same physical process. But yeah, I think that's right, that it's that at the carrier densities that we're at, they effectively, if you were to look at the field in, in our simulations, we see this in the simulations too, if you plot sort of the field along the wire, it goes to 0 where the photo-generated carriers are. And it's because of this screening or effect. They're sitting on glass or quartz substrates in air under ambient conditions. So we have looked at a little bit. We've now started to, with Jim's group, look at sort of different passivation. So they can like grow the nanowires, and you could have a native oxide on it. Or they could grow a thermal oxide on it, or they could do hydrogen annealing and different surface treatments. And we have started to look at comparing, different surface treatments. And there is a difference in the kinetics when you do that. That the thermal oxide gives you longer lived carriers than sort of when you do native oxide and so on. But we haven't done that sort of systematically yet, and we're going to start doing that. Uh-huh. Yeah, so there could be like chemical contamination. Uh-huh. Yeah, yeah. I think that there's right. That's right. And Jim and I were talking, and he said, well, if you want a really good surface, then you just need to put them in HF, and because that gives you like the best. And I'm like, I'm not going to put that on top of my objective, and So yeah, we are starting to think about things like that, but at the moment we're not really sort of equipped to kind of do that. The other direction I think we're really pushing to go is low temperature. So now we got a cryostat, we can go down to like 10 Kelvin or so, and we're sort of building a microscope around a low temperature cryostat, so we can do the experiments at low temperature as well. That's right. That's right. That's right. And so there's a lot of interesting things that we could be doing in those bends. Yeah. No, that's right. And I think we're just starting to. The experiments I showed you on the bent wires are, just a couple of months old, and we're starting to now think about what can we do in these materials under spring. Yeah. Can we image the depletion region without? So we can do. So with just one pulse or the other, about all we can do is just sort of transmission imaging. We can measure like the change in intensity of the beam as it sort of scatters or is absorbed by the sample. And in those types of images, the wire, there's no, they look uniform along the wire. Because there's no, in silicon, there's no fluorescence to sort of, grab a hold of or anything like that. So about all we can do is just sort of transmission imaging with just one beam or the other. We have, so the samples we have like on a registered, like a grid. We make we used to do it with photolithography, now we do it with electron beam lithography. But it just gives us sort of a grid, a marker pattern that we can use to find, that we can see in the optical microscope and in the SEM. I think there were kind of two. I mean, one was the bent wires. I was kind of, I mean, in retrospect, it's not that big of a surprise, but you know, at the time, I was like, oh, they're gonna, you know, they're all just, they're just bent. I mean, it's not. And so I think we're gonna be pushing on that and trying to maybe do this experiment where you sort of stretch them and but a little bit, maybe a little more sophisticated about it, so we can do it in a more controlled fashion. I mean, right now we just kind of go, you know. The other one was in the end was not really so much a surprise in retrospect, but was that how much the carrier interactions, you know, how much the number of carriers that we create sort of affects the fields and the things that are happening in the wire. And so one of the things we're going to try and do is go to much lower excitation intensity, so we'd be at much lower carrier densities. We'd probably have to decrease our pump intensity by, I don't know, it could be, you know, 3 orders of magnitude to kind of get down to that level. And so optically, that's going to be pushing it, I think, if we do optical detection. So one of the things we're thinking about is electrical detection, where you'd actually wire it up to a circuit and then actually detect electrons that are generated as opposed to photons. And there have been some people that have done stuff like that, and it looks like you can probably, you know, might be able to do much better in terms of your sensitivity that way. So that's kind of another direction I think we'll probably be moving in as well.
Did you check whether the sensitivity to the photon energy of the probe in terms of the role of interband transitions and things of that sort?
The photon energy being the color, like the color of the probe, or the yeah. So yeah, in the microscope, it's hard to do that. It's all single wavelength, and we do have in these particular experiments we weren't using our OPO. We were using like the pump was formed from the second harmonic of the probe, and so they were sort of locked. Okay, so in the microscope experiment, the answer to the question is no, not really. In the transient absorption experiment that I showed you, all of that data was collected with a broadband continuum probe. And so in these nanowires, you have in the near infrared, there's a very broad bleach that extends across, you know, from about 500 nanometers out to, you know, about 900 or 1,000 nanometers. And for the most part, its kinetics are all, it just decays sort of uniformly. It doesn't change and shift. And so I think any probe wavelength that you pick, you get roughly the same kinetics. We do see in the transient absorption experiment, we see actually that bleach grows in slightly at first and then decays, and we think that we're actually seeing the decay of the carriers as they sort of rattle down to the bottom of the conduction band. And that takes about, the lifetime of that's like a few hundred femtoseconds. That's what we saw a growth of about 300 femtoseconds, and we assigned it to that. But we haven't sort of observed that in the microscopy experiment. You mean from like the gold, or so? Yeah, they're actually. So in these images here, in this experiment here, like the pump and the probe are being dragged over the sample. So out here, this would be essentially what the signal would look like from the gold catalyst. And so initially, you see a very strong bleach that decays very quickly and becomes an absorptive sort of signal. Now, is that absorptive signal, that could be scattering from that gold particle, which would look like an absorptive, you know, sort of, yeah. But we haven't really looked into like what is that and why is it there. But yeah, but we do have some data in that, looking at the catalyst. Yeah, so that would be really cool. So we've gotten a lot of samples from Jim where they have something happens during the growth process and it becomes unstable, and all of a sudden it starts growing in a different direction. And they're so far in Jim's group, it's been more of an uncontrolled process, but there are people that are really good at it, and there are things in the literature where it looks like, a zigzag, sort of structure, and those would be some really interesting things to look at, sort of how the carriers behave in and through those regions. Oh man, that would be so cool. So yes, Jim's group can he's my best friend, and he has this ability where he can take one of these mats and collapse all the wires, and then fuse the wires through a process where they basically looks like spot welds the wires together. We've started looking at those structures, but the carrier recombination in those junctions is really fast, and that becomes a problem because if the carriers recombine, they don't have time to move anywhere, and so we've tried that, but we're still trying to get samples where the carrier lifetime is long enough that you could actually observe it. Now Jim says that they can do electrical measurements and they can see that they are actually electrically connected based on resistivities and things like that. So in principle, it should work, but we're going to have to do some work on the carrier lifetimes to get to the point where we can see it.